Well, good morning, good afternoon, and good evening to all attendees here. Thank you very much for taking the time to join us. We are located here in Madison, Wisconsin, coming live to you from DNASTAR headquarters. My name is Dr. Tom Lynch. I'm joined here by Dr. Steve Darnell, who is our team lead for all of our protein application development. Before we jump into the NovaCloud services and the description and a live demo of all the Nova applications, I just want to start off with a brief overview of DNASTAR and what we're focused on here in software development. The main focus in our development efforts are based on building workflows for the molecular biologist. Today's molecular biologist is being asked to do more, when you're looking at anything going from nucleic acid to protein to pathway analysis. And so we're building within our software efficient workflows to support commonly used lab activities such as in silico cloning, experiments, running multiple sequence alignments from both gene to genome, being able to assemble both Sanger and NGS data in order to identify any variations within your sequences, also providing visualization tools and analysis methods to look at multiple data types for complex NGS analysis, along with database access so you can look at reported variations across the world of research. And more recently, we've focused on protein workflows in order to generate protein structural models directly from sequence, antibody modeling, and also being able to simulate docking of two proteins in order to, provide the life scientists with more power and efficiency and access to tools to help their research. A little bit about our software and the focus of our development group. Number one is accuracy. We want to provide the life scientists with the best starting point for their research. So accuracy in the development of our tools is our top priority. The next is efficiency. We all know that the time in the lab is very valuable and your time is short, and that having access to the best answer in the shortest period of time is very important. So we're constantly developing our software and improving it, not only to build upon our accuracy, but also the efficiency and the time required to go through complex calculations. And then finally, resources. We want to make sure that being able to run complex calculations, you shouldn't be limited by the hardware resources that you have. So we've developed algorithms that take advantage of, for instance, hard drive space, which is very inexpensive, and more recently developed tools that can be accessed on the DNASTAR cloud hosted by Amazon, so you can tap into resources when, say, local hardware resources are running short. A little bit about our software. Our software is comprised of 3 suites of applications. The first one is the molecular biology suite. And this set of tools allows researchers to do sequence editing and annotation, multiple sequence alignments, cloning experiments, as well as Sanger data assembly and analysis. With the advent of next-generation sequencing 10 or so years ago, we started to develop tools to support all NGS workflows from RNA-seq to ChIP-seq, whole genome sequencing, and not only just the analysis of it, but also developing very powerful tools for the fast and efficient and accurate assembly of the large quantities of data that are currently being generated by tools from Illumina or Ion Torrent or PacBio. So we're supporting those workflows in a fast and efficient manner. And moving on to today's topic, our structural biology suite. A few years ago, Steve and his team started working on the foundation for our structural biology suite, and that is our molecular viewer shown here. This tool is called Protein 3D, and this is where you bring in the data from PDB or other resources to do analysis not only on the linear sequence of a protein, but also the three-dimensional structure of a protein. And over the last couple of years, we've brought in a couple key pieces of IP that were the foundation of our new NOVA applications. And these are the NOVA applications that feed the molecular viewer. The first NOVA application that we're going to present today is NovaFold. This is based on the I-TASSER algorithm out of the University of Michigan. This is for protein structure prediction direct from linear protein sequence. NovaFold Antibody, which is a homology modeling application that will generate models of antibodies from sequence using a collection of template libraries, as well and finally NovaDock, which is our protein-protein docking application based on the SwarmDock algorithm from the Bates group at the Francis Crick Institute. And all of these results then feed into the Protein 3D molecular viewer. A little bit more about the NOVA applications themselves, starting with NovaFold. We're fortunate to form a strong partnership with Dr. Yang Zhang at the University of Michigan, where he's been developing the I-TASSER algorithm, which is a hybrid protein structure prediction algorithm which uses a combination of protein threading and ab initio folding techniques. This is proven to be, in competition, to be a very highly accurate structure prediction algorithm, and we have since brought in the algorithm and formalized it and commercialized the code to provide our customers an automated hybrid approach that we have now branded as NovaFold. In order to make sense of the result of these predictions, we have generated very easy-to-use reports. It'll help guide the end user towards using the best structural model based on certain statistics, so they have a good starting point to then do further protein engineering or construct design or mutation. At DNASTAR, with all of our applications, and certainly here in the protein field, we're always striving towards continuous improvement. We want to make sure that we can always build on the highly accurate structural models that we generate and continue to improve on accuracy. Just to kind of give you an idea of the performance of the underlying algorithm for NovaFold, the I-TASSER algorithm in competition has proven to be top in class for all automated services. In the last 5 CAST competitions, which is the critical assessment of structure predictions, the Zong server, which is automated I-TASSER, has shown to be and proven to be the top in terms of the rating that they go by, the Z-score, when compared to groups of 20 or 30 other automated structure prediction algorithm and workflows. Moving on to NovaFold Antibody. This is a little bit different than NovaFold. NovaFold is a very computationally intense program. Structure predictions can take up to 10 to 20 hours just due to the semi-de novo approach to the modeling. NovaFold Antibody is strictly a homology modeling program where it takes advantage of a library of antibody frameworks for the constant regions in order to model Fabs or heavy chains or single domain antibodies. As you can see here on the right, we have kind of a brief schematic of how NovaFold Antibody works, where it's searching for framework templates, it's doing alignments, it's also grafting CDR loops. We also pay special attention to the H3 loop. As most of you know, H3 is a key determining region on an antibody for specificity and partial affinity against its antigen. And so recently in June, we released a new version of NovaFold Antibody where we are making improvements in H3 loop modeling by using a fragment-based approach where we're sampling all loop structures from nature and letting kind of nature guide towards the best model of this H3 loop. And what's nice about this NovaFold antibody process is it'll actually automatically annotate your CDR regions for you, both on the heavy and light chain. So you have a quick guide and quick access to kind of the key areas and regions of an antibody structure. This is an automated process. It will choose the templates to build your models for you. You don't need to do any of that hands-on. We'll kind of get into more the launching of structural predictions shortly, but you'll see it's very fast, easy, and automated. Moving on to NovaDock, where the underlying algorithm for this application is the SwarmDock program developed by Dr. Paul Bates and his group at the Francis Crick Institute in London. This is part of a newer class of high-resolution docking algorithms where protein flexibility is really explored during the docking. This is truly a de novo way of doing docking where you're sampling around the ligand or receptor from equidistant points and running hundreds upon hundreds of potential swarming, we'll call it swarming, docking onto the ligand or receptor, whatever you identify, whichever protein you identify as the receptor or ligand. In competition, this has proven to be a top performing tool in the pre-competitions that have been held a couple of times. It's not, it's more of a newer program for DNASTAR, and it's been a wonderful relationship that we've had with the Bates Group in continually to develop this program in taking the scale or the time required for predictions from hundreds of hours down to 12 or 13. So again, kind of like with NovaFold, calculation, it's a little intense in terms of time required, wall clock time, but when you look at the accuracy of the result compared to other known, say, crystal structures, it's well worth the time required to wait for the model predictions. And as with, we'll get to the result in a second. As I was saying before about taking into account available resources, we actually provide access to these NOVA applications two different ways. One is through the molecular viewer itself, Protein 3D, where you can log into your DNASTAR account directly in-app and launch predictions on the DNASTAR cloud hosted by Amazon. Or you can go to DNASTAR.com, register for an account. Your first prediction is free. Or you can do single purchase of predictions or sign up for a term license where through the web app you can submit your sequence, whether it's NovaFold or NovaFold Antibody, or submit your .pdb files for a NovaDock prediction, set advanced parameters, submit the job, and be able to take advantage of the cloud resources to generate these models. I'm sure everyone can agree security is very important when dealing with sensitive sequence information. We take that very seriously as well. The process in which our customers access our NOVA applications through a secured login, data transfer is secure. The NOVA applications themselves run on secure AWS virtual computers, where then the result is stored encrypted and is able for download through a secure login on your DNASTAR account. little bit about the output. As I said before, we're trying to guide the end user towards the best result and give them the statistical analysis to help guide them so they have the best starting point. Within each of the Nova apps, there are reports. I'm just giving you a quick highlight of the NovaFold output, where we present information such as the top 5 models along with quality measurements, looking at TM score, RMSD, providing you with information on what threaders were used and the template details that were pulled from a non-redundant PDB database, as well as then giving you very efficient one-click access to related structures and structural alignments, and even sequence alignments in our molecular biology tools. Continuing on with the NovaFold output, throughout the NovaFold prediction, it also runs through calculations to predict protein function. If you have a sequence of a protein of unknown function, it will determine or make a determination based on fold recognition of predicted function, whether this is an enzyme that has a turnover capability of a small molecule. It will make that prediction and then give you links to related enzymes. It will actually model in small molecules into the predicted active site and make estimations of what residues comprise an active site, and also give you direct links then to Amigo and gene ontology and potential function. Moving on to NovaFold Antibody. This output is a little bit more straightforward. As you can see, we provide you clearly with the model of the protein itself, the annotation of the CDRs shown there in the features panel, and then on the right, we're providing you with information about the coverage and the templates that were used to generate this. In this case, this is a fab comprised of partial heavy and light chain. NovaDock. This is a unique report in that this generates up to 10 models of potential docking poses between ligand and receptor. On the top left in the model overview, and if you can't see it too well, we'll get into the live software and it'll become a little bit more clear. But as you can see, there's little bubbles surrounding the receptor, and in this case, this is a fab fragment. So the ligand center of mass is represented by these spherical representations. And like with a stop and go light, green is good, yellow caution, red is bad, stop. Not only do we base, based on energy, this kind of color coding, but also the cluster size within the swarm and how many times something attempted to dock there in that interface. The size of the sphere plus the color trains the eye, very quickly into what you should be focusing on based on the statistics. As you go through the report, you see the 10 different models, and the spherical representation of the ligand now turns on into the actual ribbon model highlighted in yellow right there. So that's model 1 open. We'll go through the report itself in the live software. You can kind of take a look and you see not only do we turn on the interfacial contact residues, but we also provide you with information in a tabular data format to make your identification of what side chains are thought to interact with what from each receptor and ligand very quickly. In all, our applications not only being built on accuracy and efficiency and resources, this together, along with the solid science and the IP that we've been able to generate in-house and innovation and IP that we've been able to license through strong partnerships and collaborations, we're building tools where we make it easy to learn. The solutions are integrated between molecular biology and genomics and protein-focused workflows. These applications require modest hardware requirements, and we have PhD level scientists on both our sales and support team to provide technical support either by phone, email, on-site, webinars, chat, videos. So before we go to a live demo provided by Steve, I'm going to exit out of the presentation and launch our website. Just to give you an idea of how easy it is to start a structural model prediction. So if I've logged into my account at DNASTAR.com, as you can see, you get information about the representative from my sales team. Shown here, you have information about the licenses that you own, genome templates and transcript annotation database. Those are online tools that support our genomics workflows. And we go down here to NovaCloud services, which provides you access with NovaDock, NovaFold, and NovaFold Antibody. And as you can see here, if we want to start a prediction, we just simply click on one of these buttons. We also, within your license page, provides you access with any result of a prediction that you've run. Here you can see I've run a NovaFold prediction. I can then download this file for viewing in our Protein 3D molecular viewer, or I can simply start a new prediction. Where if I go to NovaDock, and all these, the 3 pages are very similar. I'm just going to go through this one, but you have the ability to select a receptor, enter in your .pdb file, select your ligand, enter a prediction name, set advanced parameters in terms of how intensive you want the calculation to be. If you have any knowledge, in this case, of NovaDock, of what side chains or contact residues are in close proximity between ligand and receptor, or at least should be, you can kind of guide the NovaDock algorithm towards docking in that specific areas, and then you submit the job. So it's pretty easy to get started. Once the result is done, then we can go into the Protein 3D viewer, which I've done here. And I'm going to hand this over to Steve, and Steve's going to go a little bit more into some of the more technical details and information that are provided in the reports. Today, as you can kind of see going through our molecular viewer, we have multiple projects open, very easy to access in this tab format in this window. But we're going to start with a NovaFold prediction, move on to a NovaFold antibody modeling problem, and then go on to a NovaDock experiment and compare that to the actual co-crystal structure of an antibody antigen. So Steve, I'll hand it over to you now.
All right, thank you, Tom. Before we kind of get into viewing results from our different NOVA applications, I thought I would kind of give you a brief overview about the Protein 3D application. This is our molecular viewer, as Tom has said. To guide our trip through some of the features of it, I'm going to use one of our features, and that is you can open files directly from the protein data bank as well as files from your local file system. I'm just opening up a myoglobin structure just for something to talk around. So by default, when you view a structure, you're going to see these ribbon diagrams with any ligands available displayed automatically like this. Our Explorer view provides an overview of what molecules are associated with the structure. In the structure, of course, protein chains or nucleotide chains as well. They will be listed here, as well as any ligands that might be present as well. Throughout the entire Protein 3D application, we have an idea of selection being shared across all aspects of the documents and side panel views. So making a selection in one place will show selections in our sequence view, in our details view, our Explorer view, and others as well as we get through them. In terms of what you can control in terms of molecular styling, we have a number of options in terms that you can control different rendering styles for different groups of the molecule or of the structure, the backbone itself, side chains, ligands, and water molecules if present. Additionally, for all structures, you can calculate surfaces. You can choose to calculate a surface using the entire structure if you like by not selecting anything, or if you are more interested in other parts of the molecule, you can make selections as well. Those renderings too can be controlled here for surfaces. You can change between molecular and solvent accessibility and different sorts of renderings and transparency as well for surfaces. Colors are controlled through the same side panels. Additionally, we have a view for managing and interacting with feature annotations that are found in the structure. The protein data bank comes with a number of secondary structure and ligand binding site annotations, which are presented here. You also have the option to create new features based upon regions of your interest, and you can make a selection and create features from this panel here. And you can come back and then edit them or interact with them later so that you can highlight regions of interest so that you can come back to them later. Our details panel is usually something I recommend that stays open while you're interacting through Protein 3D. It will change depending on the context of the selection. In this case, since nothing is selected, we're showing information about the overall document, in this case a structure. But as you select features, you will get different information regarding the regions that are being selected. You can select individual residues and receive information like biophysical properties of this selection, as well as some of the structural information like dihedral angles, phi, psi, and omega angles. You can also, through the details panel, you can look at distance calculations between pairs of residues. So all you have to do is select two residues, and we will show you the distance between the two closest atoms associated there. And in the cases when you have surfaces created, we will also tell you what or how much of the surface that the residues in the selection contribute to the overall surface area that you had created. We also have a full suite of bioinformatic sequence analyses that can be applied to the molecules in your structure. Biophysical sorts of analyses from hydrophobicity, different types of stability metrics, transmembrane predictions, and the like listed here. We also have immunogenicity sorts of metrics, including antigenicity, a B-cell epitope prediction, a linear B-cell epitope prediction method that we made in-house, and a few others, as well as some database-style predictions, as well as like a large set of proteases that you can decorate your protein sequence with, as well as access to the ProSite domain and site library or database as well, and some protein modification methods as well. Again, all aspects of the analysis view are selectable as well, so you can make selections for regions inside the analysis view. In this case, I selected what we were predicting to be one of a couple of strong linear B-cell epitopes. If we were to look at that in other views, we'll see that same selection being represented in sequence and in structure as well as the details panel. So that's just kind of an overview of some of the capabilities just that our molecular viewer can do, but our NOVA applications are designed to create new data for presentation inside of Protein 3D. The first document here is the result of a NovaFold prediction. This is an aldolase from a microorganism called B. macroti. This example is kind of interesting because there actually are no known structures from this organism in the protein data bank. And it highlights that you do not have to have extremely high sequence identity between structural templates and your query sequence in order to generate high-quality models. In this case, this aldolase sequence here really only has kind of moderate level sequence identity between other templates in the protein data bank. That 40 to 60 range is what's being presented here. So often homology modeling tools, generic homology modeling tools, function very well at 70 and 90% and at those higher levels, but it gets more difficult to identify sequence structural relationships at like less than 50%. NovaFold is unique in the fact that it doesn't use sequence alignments directly in order to select templates in sequence homology. It uses protein threading. Protein threading is different because not only does it consider sequence similarity, it also uses predicted secondary structure and solvent accessibility and predicted internal residue contacts. So we're using structural information, in this case predicted structural information, but still nonetheless, in addition to sequence information to generate these alignments. And this is what allows us to get to identify statistically significant templates with lower and lower sequence identities. In this section here, we show the top 10 templates that were selected from by NovaFold, including identifiers, scores, threading information, but also for summary, visual summary, like the percent coverage that the template has over your query sequence, and also the sequence identity percentage as well. And much like in other places, as Tom had suggested, we use stoplight colors in order to help identify different levels of fitness between, for different types of information. These template mini-maps are colored here based upon sequence identity. So it's just an indication of one more quality metric, but again, it doesn't tell the entire story. You have the ability, too, to select multiple templates, and you can choose to automatically open models that were created by NovaFold and align them against templates too, if you so like. Our model overview gives a quick summary of up to 5 models that are predicted by NovaFold, including summary metrics regarding the expected quality of a structure prediction. We present 2 types of information: a predicted template model score, TM score, as well as root mean squared deviation, RMSD. TM score is typically the metric that we have people look at when giving a quick quality overview of the different models. TM score is a normalized metric comparing 2 protein superimposed protein structures. The scores here are regression scores based upon a statistical model to be a measure of confidence. So the closer that this TM score, predicted TM score, is to 1, the better. So 0 is worst, 1 is perfect, and anything above 0.5 is predicted to be that the fold itself is at least correct. Although around a TM of 0.5, you're going to have less confidence in the details of like the external loops. But the core fold would still have a much higher degree of confidence. Each model has an interactive structural thumbnail that you can interact with, including some of the summary information. Each one of these can be opened up into a full Protein 3D structure document. We'll show that in a little bit. We also have the additional option of trying to predict small molecule binding sites and protein function based upon these models. So what's unique about this workflow is that we're not using sequence to make these predictions. We are using the structural model itself, and we're doing basically a reverse structure search throughout the protein data bank, looking for similar experimental structures, and then using these structures and the binding sites that are contained within them to effectively do a data mining operation to predict small molecule binding sites, such as that are listed here, as well as enzymatic sites and protein function. I'll start with protein binding sites. Again, these thumbnails are interactive, and selections within them will affect your structure as well. We also list the binding sites themselves, moving down to predicted protein function. We also use Go terms, or we try to predict Go terms that are associated with a query structure as well. And in this case, with this aldolase, we're starting to see the sorts of markers we would expect for a high confidence model. So we're seeing fructose bisphosphate aldolase activity and fructose binding activity. With, again, a 0 to 1 degree of confidence, we're seeing 0.9, 0.8, the high levels of confidence. So this is yet another way in order to query the quality of a model in a way that is not strictly regarding just the fold itself. We're trying to use the function as well. When you have a model that you're interested in interrogating further, you can open it in a full structure document, and then you have all of the capabilities that we had described before in order to interact and to interrogate your structure. Additionally, information or models that are opened from the NovaFold report. Well, actually, if we were to have opened models from down here, we would actually have this binding site information be annotated automatically in the structure documents as well. Models that are opened from up here are just the model itself. Moving forward, this is a result from NovaFold Antibody. Again, as Tom had mentioned, NovaFold and NovaFold Antibody are very different algorithms. NovaFold is a general purpose tool that uses ab initio style techniques and can take several hours or maybe tens of hours to complete a simulation or a prediction. NovaFold Antibody is more of a homology modeling by parts approach, as that workflow that Tom had shown. We're looking for we're selecting framework regions for heavy and light chains independently and assembling them. We are grafting canonical CDR loops, with the exception of the H3, onto this structure. We're using natural loop structures extracted from the protein data bank, from both antibodies and from all structures from the protein data bank, looking for things that nature has identified that could be useful for modeling an H3 loop. When you open an antibody document, you will actually get this annotation occurring automatically, where we will identify the hypervariable regions and the different loops as well. And since these are just top-level features in Protein 3D, you can select them. You can see information regarding that particular region. You can also identify we'll annotate where the disulfide bonds can occur with larger antibody constructs as well. Now, modeling an antibody structure is all fine and good, but we have more ideas about how we want to use this. It's not just about getting an antibody structure in its unbound state. We want to use it in order to do things like predict epitopes, conformational or otherwise, against antigens. And we're interested not in just doing it through a sequence-based approach, by using biophysical models through docking to help make these conformational epitope predictions. So this leads into NovaDock. We're using, in this particular example, an antibody structure paired against an antigen structure. Now, the terminology used in NovaDock and other docking algorithms is typically receptor and ligand. In reality, these are just conventions. You can make anything you want to be the receptor or the ligand. In this particular case, we've made the antibody structure, Fab structure, to be the center of the universe of our docking, the receptor. Binding partners do not have to be single chains. They can be multiple chains like an antibody. So a heavy and a light chain are represented here as the receptor. The docking, after the docking is done, actually, we hide this just for convenience so that we don't show too much information. But here, our receptor heavy and light chains are presented here. And here, this is a neuraminidase molecule bound to an anti-neuraminidase Fab protein, or IG fragment. So these are the two inputs to our NovaDock prediction. This is the summary that Tom had referenced before. Again, an interactive thumbnail that you can move around and view in order to get a good view of what you're interested in. Here are these heat bubble spheres identifying where the top 10 models actually co-locate in 3D space around our receptor. Selections in the table, highlight them in the thumbnail as well. We also use the green to red coloring in order to highlight lower energy scores as being our most confident poses, and higher energy scores to be less confident. And these spheres too, the radius of them is proportional to the cluster size as well. Typically in protein docking, we are sampling hundreds of final pose conformations, and we cluster them by geometric similarity. When clusterings become greater, there is a higher sampling. The algorithm starts to, with multiple predictions, starts to focus and converge on particular regions of the patches on the receptor. So larger cluster sizes are often another quality metric indicative of better models. When you look down further, you can get some summary information again regarding our models 1 through 10. This one is our top model here. Again, this interactive thumbnail now shows the full atom representation of the interface as well as the full ribbon structures of both ligand and receptor. The same information, summary information, is located here, as well as a table that describes intermolecular contacts for every interface residue between the receptor and the ligand. So we annotate which residues are part of the receptor, which are part of the ligand, and we also tell you which residues on the other binding partner are being being in contact with these residues as well. Selections in this view, or in this table, again affect selections in the thumbnail view here. You can open these models again in large, or in the full Protein 3D document, and we've done a little bit of structural analysis with model number 1. So we opened model 1, again, this blind docking simulation between this Fab and neuraminidase as this antigen. And since we happen to, in this particular case, the structure of the two things bound together was already solved experimentally. So we've performed an alignment between our model, which is in color, the yellow, green, and blue, and then the experimental structure, which is in gray. One feature that you can use in Protein 3D, which I didn't actually show off, is that you can actually open groups of structures all at once, or you can add new structures like we did here to an existing document. You can perform structural alignments. If you choose not to make any selections and just select one of our two alignment algorithms, we will attempt to align all structures against the first entry in this list. You also have the option, if you so want, to select and direct the alignments as well if you had wanted to. So we could choose to align just the heavy chains against one another, and then the rigid body rotations would be propagated through the entire structures based on that alignment. The root mean squared deviation, the RMSD, between these two structures, another metric of protein similarity, structural similarity, is reported here. RMSD is an average distance between all of the pairwise superimposed residues between two structures, which means that since it's a distance metric, the closer to 0, the more similar two structures are. In this case, we have an RMSD of approximately 0.4. So what that actually shows us from this calculation and visually is that our NovaDock prediction for this model, from this model 1, has a really high degree of similarity between the experimental structure. So this is just an example of what you can do with protein docking. And not only that, the features from before are annotated here in the site residues, so you will also be able to interrogate what are the residue, the interface residues that make up the conformational epitope by selection here. Or conversely, if you're interested in the paratope for this particular example, you can select the receptor contacts and get a listing of all the residues that are involved there. So that's kind of wraps up our overview of the Nova apps, all three of them doing structure prediction, both for general purpose as well as antibody fragment modeling and protein-protein docking, and how we use and visualize that information in Protein 3D.
Thanks, Steve. And we have a few questions that have come in from our attendees today, so I'll go ahead and pose those, and I don't know if you want to answer them, Steve, or if Tom Lynch wants to answer. Yeah, we'll just see. The first question is, how does licensing work?
Yeah, so I can take this. Licensing is pretty straightforward. As I mentioned before, your first prediction for our Nova applications is on us. After that, you have the ability to purchase predictions or sets of predictions, as well as annual licenses of each. They are licensed separately, so it's at DNASTAR we're very accommodating, and we just want to make sure that we find the best licensing solution that meets both your research and budgetary requirements.
Thanks, Tom. Next question is, after a NovaFold prediction, can you make mutations to the side chains? And then a related question, how do you compare wild type to mutant structures?
Well, I'll be happy to answer that. In the current releases of NovaFold and NovaFold Antibody, the way that one can model mutations is by performing independent structure prediction processes. So you would basically provide mutant sequences and wild type sequences and run a full prediction. And then in terms of analyzing or looking at the differences between those, you would again open the results together, the models together, into a structured document and perform an alignment. In our fall release, though, that's going to change, and we are going to be providing a new tool for doing mutations directly within Protein 3D. You'll be able to make a selection and select a mutation that you're interested in making, and we will go and model one or a small set of mutations and kind of do a little bit of repacking of the local environment and report a statistical metric whether or not we think that the mutation is going to be disruptive or indifferent or perhaps somewhat stabilizing as well. So that is going to be happening in the fall. We will be providing user experiences after that where we'll automatically do any alignments that need to happen, show any side chain overlays that you'll be interested in as well. And that should be done automatically so that you won't have to manually do it. You can get to your work a little faster.
Great, Steve. Thanks. That sounds like it's going to be a really useful feature in 15.0. And the last question that we have is, what's the difference speed-wise between using the I-TASSER server and the NovaFold Cloud?
Yeah, I'll kind of start, and maybe Steve, you can fill in some gaps if there are any. For all intent and purpose, they're very similar. As Steve and I have been mentioning, for NovaFold predictions, they can take upwards of 10 to 20 hours. This is pretty similar to the I-TASSER server. We have gained some efficiencies in parts of the code that Steve maybe can expand on a little bit to help improve time. But in the grand scheme of things, 10 and a half, 10, 11 hours is, there's not a lot of difference there. They're both still kind of overnight. Where we do gain the advantage, though, is in terms of access. We do not have a queue for any predictions to wait for. Also, we have the ability to open up resources to allow many simultaneous predictions. We, as researchers, use our NovaFold application. We have two kind of levels of users, those who run a single or maybe a small handful of predictions at a time, and those who are doing hundreds of predictions at a time. So, through licensing and through cloud resources and local resources, we have the ability to allow researchers to do many simultaneous predictions. Steve, do you want to?
Sure. In terms of some of the technical differences between NovaFold and I-TASSER, the differences are more in terms of computational efficiency. We went through great pains to make certain that the the results are the same quality, nature of results that you would get from I-TASSER, but we have re-implemented the main simulation algorithm or program that is in use in order to make it a little more numerically stable, as well as parallelize it as well. So we, because we'll be on the cloud with our NovaFold Cloud predictions, we can bring and instantiate multiple machines, and we can have multi-threaded simulations running. So again, we'll be able to cut these very long, like, what could be 50-hour simulations if they were to be run kind of in a serial process by factors of 8 or 12 or whatever we want to, based upon the machine types that we bring up, and being able to use the ability of Amazon's cloud to scale horizontally to accelerate your predictions.